Hi, my name is Nurafika Najwa, and today I will be presenting my poster entitled Evaluation of Bioelectricity Production from Sagu Processing Wastewater using a co-culture of Clostridium butyricum and Geobacter sulfur reducens in a microbial fuel cell. Sagu, or also known as Metroxylon sagu, is an important economic crop. However, sagu processing generates a large amount of wastewater that are rich in organic matter. If discharged without treatment, this wastewater can increase chemical oxygen demand or COD and biological oxygen demand or BOD, which may contribute to environmental pollution. One promising solution is the use of microbial fuel cells or MFC. These systems utilize microorganisms to convert organic matter into electrical energy while simultaneously treating wastewater. In this study, a co-culture of Clostridium butyricum and Geobacter sulfur reducens was investigated to improve both electricity generation and wastewater treatment efficiency. This study have three objectives. The first one is to evaluate the potential of sagu processing wastewater as a substrate for bioelectricity production in a microbial fuel cell. The second one is to investigate the performance of a co-culture of Clostridium butyricum and Geobacter sulfur reducens in enhancing electricity generation and substrate degradation. The last one is to assess the feasibility of the system for sustainable wastewater treatment and energy recovery. For the methodology, sagu processing wastewater was collected from a local sagu mill and its initial pH, COD, and BOD values were measured. The bacterial cultures, Clostridium butyricum and Geobacter sulfur reducens, were cultivated anaerobically at 30 degrees Celsius. Equal volumes of both cultures were then mixed to prepare the co-culture inoculum. A double-chamber microbial fuel cell was assembled using carbon electrodes and a proton exchange membrane. The bacterial inoculum and wastewater were introduced into the system, and voltage production was monitored using a digital multimeter. COD and BOD measurements were also conducted before and after treatment to evaluate substrate degradation. All experiments were performed in triplicate, and the data were analyzed using one-way ANOVA with a significance level of P less than 0.05. Moving on to the result, the co-culture of Clostridium butyricum and Geobacter sulfur reducens significantly improved electricity generation in a microbial fuel cell. In addition, reductions in COD and BOD levels were observed, indicating effective degradation of organic matter in the sagu wastewater. Further discussion, the low electricity production observed in the Geobacter sulfur reducens culture suggests that this bacterium alone was unable to efficiently utilize the starch-rich wastewater as its carbon source. In contrast, the co-culture system showed improved performance because Clostridium butyricum was able to break down the complex starch present in the wastewater into simpler compounds such as acetate. These compounds then serve as a carbon source for Geobacter sulfur reducens. Geobacter sulfur reducens could subsequently utilize the acetate and transfer electrons to the anode through the formation of an electroactive biofilm, leading to increased electricity generation. The synergistic interaction between the two microorganisms enhance both substrate utilization and electron transfer efficiency. Furthermore, the reduction in COD and BOD values indicates that organic matter in the wastewater was effectively consumed during microbial activity. Therefore, the system not only generated bioelectricity but also contributed to wastewater treatment. In conclusion, the co-culture of Clostridium butyricum and Geobacter sulfur reducens improved electricity generation and reduced COD and BOD levels in sagu wastewater. These findings demonstrate that sagu wastewater can serve as a suitable substrate for microbial fuel cells and that combining fermentative and electroactive bacteria can enhance overall system performance. Overall, this study highlights the potential of microbial fuel cells as a sustainable approach for simultaneously wastewater treatment and renewable energy production. That's all from me. Thank you.